Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Preclinical Models for Microbiome Research in Oncology and Inflammatory Diseases. My name is Sonia Hunt and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to send your questions and comments to our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Oncodesign, who developed the content for this presentation. Now it's my pleasure to introduce your speakers for today's webinar. And first, I'd like to introduce you to Sylvie Maubant. She's a head of study research unit in Vivo Sciences Department with Oncodesign. Sylvie has more than 15 years of experience in preclinical research in the oncology field. She obtained a PhD in biology, medicine, and health from the University of Caen in France, and then she continued her career as a postdoctoral researcher at several institutes to work on projects related to identification of new therapeutic targets, drug development, and/or validation of therapeutic tools in collaboration with industry partners. Next, it's my pleasure to introduce you to your second speaker, and he is Ivan Gladwyn-Ng. He is a field application scientist at Taconic Biosciences who is passionate about accelerating advancement in human health by improving the application of in vivo models and care of animals in the drug discovery process. He possesses more than a decade of experience in utilizing rodent models within many diverse fields of preclinical research, with particular interest in the studies of infectious, neurological, and oncological diseases. He has multiple co-first and corresponding authorships and successful patent applications in Europe and USA. Now, before I pass over the mic to our first speaker, who is Sylvie, we have our first poll question for our audience members to engage in. I'm going to launch it right now, and you should see it take over your screen, and there it is. Now, this is done in real time, so your participation is strongly encouraged and very much appreciated. I'll give you about 45 seconds to complete this poll. All you have to do is just select any of the bottom choices there. The question that we have is, which topic is currently of interest to you in your microbiome landscape? So we have 4 options to choose from below. Microbiome-derived therapies, if this is you, then click on that. Microbiome-based diagnostic, drug microbiome interactions, or other. So please go ahead and make your selection. And then once I close the polls, I'll share the results with everyone, and then we'll hand it over to Sylvie. Okay, that looks like it's the majority of our audience. So very much appreciative of that, everyone. I'm now going to close the polls and let's take a look at the results. Okay, so here are the results. We have 21% of our audience saying microbiome-derived therapies, then 17% are saying microbiome-based diagnostic. Then we have 50% of our audience are saying drug microbiome interactions, and then 13% that are saying other. So thank you everyone for participating in that poll question. Now I'm going to pass the mic over and the controls over to Sylvie. So Sylvie, when you're ready, you may begin.
So hello everyone. So can you see my screen,
Yes,
Sonia?
we can.
Okay, so thanks for joining this webinar involving Taconic and Oncodesign. So we will first give you a general overview of main discoveries and research data that explain the attention paid to the microbiome, and especially in a pathological situation. Second, we will present you models and services suitable for microbiome studies, and we will focus on fecal microbiota transplantation, also named FMT. And third, we will show you some results related to the FMT and obtained in rodents in healthy or disease conditions. So we will start with few definitions and notions that may be helpful for the next steps. So in fact, the microbiota is composed of a wide variety of microorganisms, including at least bacteria, fungi, and viruses. And in fact, the microbiome refers to the genetic information of the microbiota. As you know, bacteria represent more than 99% of the microbiota. So most of the studies focus on them. So finally, the microbiome studies correspond mainly to bacteriome studies. So as you know as well, the microbial imbalance, also named dysbiosis, has been reported in several pathologies. But in much cases, in fact, we are still facing the chicken or egg situation, meaning that we don't know if this dysbiosis is a cause or a consequence of the disease. In most of the in vivo microbiome studies, the animals that are used are defined according to their associated microbiota. And in fact, the holoxenic animals correspond roughly to those with a microbiota similar to the one acquired by the wild individuals. And in fact, this first category of animals can be either conventional or they can be defined and managed as a specific pathogen-free, SPF, or specific and opportunistic pathogen-free, so also named SOPF. So in fact, to summarize, in the first case, their microbiota is uncharacterized, and in the second cases, the absence of specific pathogen plus or minus opportunistic organism is checked. And the gnotobiotic animals include those whose microbiota is well defined. And in fact, this second category of animals can be deprived of all microorganisms, so are termed germ-free or axenic animals, or they can harbor defined bacterial communities. So as you know, microbiome is a new player in drug development, and in fact, efforts were made to harness the microbiome for diagnostic and therapeutic purpose, but also to understand the bidirectional interaction between the microbiome and non-microbial drugs, as it may affect drug efficacy and or toxicity. So in a pathological context, the identification of biomarkers, it refers here to beneficial and harmful bacteria and the derivatives, usually by using omics technologies, will orient the strategy to be used for shaping the microbiota and consequently the immune response to meet the current medical needs. So different approaches can be conceived for this purpose, with most that are listed here, with fecal material, defined consortium, single strain, molecules derived from the microbiota, molecules known to act on the microbiota, and the bacteriophages. And miscellaneous in silico, in vitro, in vivo, ex vivo, and human models tools can be used for evaluating and investigating these approaches in a healthy context or in disease conditions, with attention paid to some specific aspects, such as pharmacokinetics, pharmacodynamics, efficacy, and safety. In fact, whatever the topic of interest, and especially in pathological context, the clinical data have to be always at the heart of the microbiome research. For example, as a starting point to identify target of interest, or for demonstrating the benefit of a treatment after having conducted a strong proof of concept studies in preclinical models. So for example, the analysis of samples collected from cancer patients treated with anti-CTLA4 or anti-PD-1 antibodies have revealed that some microbial taxa are enriched in a context of beneficial therapeutic response, as represented by blue ribbons, and other microbial taxa are enriched in non-responders to the therapy, as represented by red ribbons. So based on that, in fact, promising strategies were developed to circumvent this primary resistance to anti-cancer therapies, here immunotherapies. And in fact, these strategies were checked in appropriate preclinical models. So in fact, back and forth between preclinical and clinical studies are required to increase chance of success during a microbiome-derived drug development process. So now we will focus on rodent models, and Ivan will present you shortly the main factors shaping their microbiome.
Yep, thanks Sylvie for introducing the definitions as well as the updates into how clinical and preclinical research in an iterative process for microbiome-related therapeutics or drug microbiome-related studies. And as Sylvie mentioned, today we focus on preclinical microbiome studies in rodents. And to better design such experiments, I like to introduce with this slide by appreciating key categories of factors that shape the rodent microbiome. So, and the microbiome, as what Sylvie mentioned, refers to the microbiology of the mouse. And it's in this first introductory slide, it's a broad overview of the microbiome status and the microbiota status of the entire animal, be they within the gut or on the fur, on the skin, or external. Later on, Sylvie and I will highlight with several publications and few case studies, as mentioned in the introduction, about specific gut microbiome and or fecal microbiota transplantation experiments. But in this case, I'd like to start with the leftmost column, which is the column of husbandry factors, because environmental influences are critical towards influencing the microbiota status of rodents. Environmental influences such as sterilization and cleaning, procedures of the cages, but also sources of the water, the diet, the bedding, as well as enrichment material. So this is key. Sometimes we overlook these factors. The third column then of now internal murine factors, where the microbiome has been reported to be a non-genetic heritable trait through the maternal microbiome, where the perinatal or juvenile period of the newborn pups has been proven and reported to be a key window for shaping host microbiome mutualism. And thus, physiological traits naturally influences this relationship, such as the sex, the age of the mice and rats, also the health and the stress if they are undergoing any, and also definitely pathological conditions such as diseases, be they spontaneously or induced in some studies such as inflammatory bowel disease, to study inflammatory bowel disease, or to study efficacy of certain immune checkpoint inhibitors. And then in the fourth column of naturally microbiology, where we're studying the microbiota. So in the mice, founder effects and drift will tend to change the microbiota and microbiome over time. So some things are known as cage effects, where vertical transmission affects the microbiota. In addition, horizontal transmission or horizontal effects due to horizontal transmission, where the microbiome is transmissible within the cage, so animal co-house, and also between cages when you transfer mice from one cage to the other. And in the last column, the sixth column, so as I mentioned, it's the environmental influences are critical, and sometimes this is key also, the humidity, the temperature of your vivarium or your barrier. And these are just the key categories that we should pay attention to when we're thinking about how to better understand the microbiota of the rodent, and then how to better plan experiments. And I'll pass on to the next slide to Sylvie.
So thanks, Ivan. So as mentioned earlier, we will focus now on fecal microbiota transplantation, and especially on practical consideration in rodents. As you know, in fact, up to now, numerous and different protocols are used Even for basic FMT procedures, such as donor stool preparation, delivery mode, dosage, duration of the administration, and the recipient preparation. So for webinar purpose, we have decided to pay attention to recipient preparation and delivery mode, as described and is summarized in this diagram. So for example, the FMT can be performed by oral gastric gavage or by enema in germ-free rodents that lack all microorganisms and are maintained in specific isolators, or in SPF rodents with the gut microbiota depletion obtained by using antibiotics or laxatives, such as polyethylene glycol. And several sampling can be performed along the experiment for validation purpose. It is to check the depletion of the gut microbiota in the recipient and also the successful engraftment of the donor microbiota. So furthermore, it is important to acknowledge advantages along the gaps and the limitations of such animal systems. And in fact, the key ones are listed and detailed in this table. For sure, different considerations come into play when designing an FMT experiment, such as translatability and also the cost. Some models, such as germ-free animals, require specialized and expensive equipment and already training, in contrast to animals treated with antibiotics and laxatives that are inexpensive. However, regarding antibiotics treated animals, there are some concerns related to the efficiency of gut microbiota depletion, considering that different cocktails, doses, and duration of treatment are proposed, and that may lead also to the emergence of resistant bacteria or outgrowth of other microorganisms present in the microbiota, such as fungi. And in addition, some injuries of the rectum due to the laxative treatment have been sometimes reported, and that may lead to an infection. So in conclusion, there is no single and perfect model for FMT, and the best approach has to be selected based on the objective of the experiment. So now Ivan will review with you the usual question addressed at time to design an FMT experiment.
Yeah, thanks, Sylvie, for highlighting the primary advantages and limitations of different models. So key questions as Sylvie has defined, what is FMT? So some people also, it's also known as fecal transplantation or gut microbiota transplantation, and it refers to the transfer, that processes that we defined here, and that's a commonly accepted definition. And that's why in our introductory poll, as what Sylvie also highlighted again, and the overview is there is no perfect model. It really relies on your aim of and goal of your experiment. And in the opening poll, what is the aim? Is it to use this FMT as a treatment, be it prophylactic or therapeutic management, comprising part of a clinical management strategy? Or is it in a preclinical setting to recapitulate the patient phenotype and to transpose a translatable as well as a valid experimental design? So in such, which is why we also asked, and we're very cognizant that it can be used to study the interaction of drugs on the microbiome and the microbiota status in mice and rats. But in both cases, there are known, well-known examples of successes. For example, the application of FMT in humans as a prophylactic intervention has been shown that by transferring the microbiota of a healthy patient to the intestinal tract of a diseased patient has been used in recurrent infection, human patients with recurrent infections with Clostridium difficile. Has also been shown with some clinical relevance in the management of patients with inflammatory bowel disease, such as ulcerative colitis or Crohn's. And in the animal studies, increasingly, there's more and more papers and publications as well as studies showing that FMT can be an important tool in microbiome research. And typically, we can think about transferring microbiotas between animals, often between mice. But actually, increasingly, researchers are referring to FMT experiments where fecal material is transferred between different species, very commonly sourced from humans, and then transferred into mice. And this, understandably, it can become increasingly complicated because in the human patient, as shown on the right, we have a lot of cells, but then, as Sylvie introduced, the microbiota comprise of not just bacteria, but others such as viruses and fungi. And all these have a complex interaction between the host, the humans, as well as the microbiota. And that's why, to now better approach the potential and designing an experiment in the next slide, there's a lot of factors to consider. And in the next slide, I categorize these into 7 major categories. They're color-coded appropriately, and then I spend some time into describing the 7 key questions or key considerations from the collection of fecal microbiota to processing of that fecal material to the storage, the choice of the recipient, something that Sylvie has introduced earlier, what type of mouse model and or rat, if you're thinking about that, as well as the others, factors 5, 6, and 7. But to spend a little time now elaborating a little bit about the collection of fecal microbiota for transplant. So when we think about that, typically researchers think, they ask, what is the method of collection? Will we perform an invasive procedure or a non-invasive procedure? And then depending on how we harvest or how we collect the fecal material, then it's related to where the site of the fecal matter is sourced from, because if we're doing a non-invasive procedure where we just collect fecal material from the bedding in the cage, or where we manually manipulate the animal to collect them, it's a good proxy for microbiota along the entire gastrointestinal tract of the rodent. But what we know is that there's definitely differences in composition and diversity between different topical niches within the gut, such as the cecum versus the colon versus the small intestine. So some researchers would do both invasive and non-invasive, and understandably, if it's invasive, training of staff comes into play, as well as you have to think if it's invasive, is it a terminal collection or non-terminal collection. Now, after you collect your fecal material, the second thing we have to consider is how do we process this fecal matter. If it's stool samples, for example, how do we minimize the impact of processing on any bias on microbial survival? And very often the key factor is exposure to oxygen, because the majority of bacterial species within the mammalian gut, either in humans or rodents, they're strict anaerobes, meaning that if prolonged or persistent exposure to oxygen will definitely affect the survival of major taxa and genus of microbes. And then after we process, and the third key consideration is how do we handle and store these to minimize or to prevent pre-bias or selection on different types of microbes. So very often, researchers will dilute the fecal matter in a media. Definitely, the dilution media should be non-bacteriostatic for most of the microbes species present. But then, how we process, handle, and store these fecal matter depends on how you intend to use them, which is in factors 5, 6, and 7 later on in the next slide. Because if you want to do a fresh transplantation, or do you want to freeze these fecal matter and then depending on experimental design, and if you're going to freeze these fecal matter, then the consideration of do we need or do we want to add a cryoprotectant? Would that affect, depending on the choice of your cryoprotectant, will it also affect survivability or recovery of different microbes? Or is just pure snap freezing of the fecal matter sufficient? Right, and all that, then we can then consider in the next slide things like what Sylvie has introduced beforehand. The choice of recipient, in this case, in this webinar, we're talking about mice. So is it germ-free or not? Or is it antibiotic or laxative treated animals? Because increasingly researchers have their own genetically engineered, genetically modified models. They want to look at them, and some of those animals may or may not survive well, or the health and husbandry may be affected by germ-free or non-germ-free status, husbandry health status in the vivarium. And antibiotic or laxative treated might also affect the progression of certain genetically engineered mouse models. And it's something that Sylvie has a lot of experience in. And then another case example, which I'll highlight later on, is the age and the sex of the recipient. Mice, be they male or female, as well as would you do the fecal, the FMT, when they're juvenile or adults. And all these are related to a lot of factors. Also, the younger you can appreciate, the younger the mouse, the more sensitive they are to manipulation, the more technically challenged it is to perform an oral gavage. And then the sex, male and females. Females would have estrous cycle. They might also affect the bidding, and so there's a lot of consideration to take to appreciate. Then, as what Sylvie also mentioned, and what I alluded to, if after deciding upon the recipient, the donor and or recipient animal model, how are we going to administer the fecal matter to? In rodents, as we all, quite a lot of us are familiar with, the primary route of microbiota transfer within co-housed rodents is typically the oral ingestion of fecal pellets due to the coprophagia. And a lot of researchers would think, okay, co-housing is a relatively simple method of transferring microbiota. However, if for increasing resolution and increasing definition and delineation of experimental design, the additional methods, all these come with many different advantages and disadvantages. And typically, we can think about them as a top-down, which means oral gastric, or a bottom-up, which is anorectal administration. And all this will also lead to effects on, if for some of our researchers today, efficacy trial, biodistribution trials. And it's just like any experimental design when we're thinking about these trials that we are performing. So next in the key consideration 6, in assessing the engraftment, when do we sample from recipients? What donors are we how do we compare them to the donors? What are the choices of controls that are appropriate to experiment? When, where, essentially when, where, how to sample. So these are also important. And lastly, safety. Never forget about the safety considerations, not just to the animals, the 3 R's, the animal health, but also to our co-workers in the lab, our vet we are utilizing human sources of fecal material, on top of fecal material being potentially biohazard and infectious, so it's standard practices for all that. But if there are human sources of fecal material, then there's also standard screening principles applied in any human to animal transplantation to protect both our co-workers in the lab as well as the animals themselves. So in the next slide, I would like to just have a brief overview of whatever I just mentioned. So when planning FMT experiments, there are many practical steps to consider. We have the limited time today, and for a brief overview, and many of these are based on current peer-reviewed literature in microbiome research, whereas a vast majority draw upon our vast knowledge of just classical microbiology. And then that is relatively fortunate. However, there's still, I think, limitations in the appreciation of either projects or publications in microbiota research, because there's a lack, still, I think, a lack of a contemporary systematic investigations of the advantages and disadvantages of FMT experiments. The description of, as mentioned, all these key criteria and key considerations, there's still some, I would say, ambiguity. And therefore, a more detailed examination of the concept described here would definitely lead to more successful and reproducible experiments in your facility, and then definitely for improved translatability as well as reproducibility for future experiments within your group. So I would like to highlight then with a collaboration in the next slide that Taconic has performed with an academic group in the University of Denmark. So we had the fortunate, we were very, I would say this is a very interesting study where the collaboration led to a very interesting finding. But we started out with the understanding of the gap in the knowledge back then in 2017, 2018 then. But that germ-free, we know that germ-free mice traditionally are colonized with microbiota of interest, and they've been used for multiple types of studies. And one of the key limitations of that, as Sylvie has introduced, in using germ-free rodents, isolators are typically used for housing such anotobiotic rodents, because there's optimal protection from the environment. But then they are very prohibitive in terms of cost as well as logistics in maintaining such an animal facility. So together with Randy Lundberg, who is a PhD student back then, but with the group in Copenhagen, we decided to assess and compare the effects of different housing systems. In this project, specifically, isolated housing versus individually ventilated cages, or IVC. And these two different housing systems on the gut microbiota composition. And on the top right, we designed a study comprising, firstly, of C57 black 6 intact mice that were the donor mice for the colonic contents. So these are pictured in black, and we utilize these fecal matter from these colonic contents from these mice to inoculate germ-free Swiss Webster mice by oral gavage. So in blue there, and then they were housed in either, as you can see, top, which are the isolator, a flexible film isolator, or just below that, in IVC cages. So in blue, they are termed P for parental because they were used to breed subsequent generations of mice housed, likewise, F1 generation in red and F2 generation in green for us in the study. And then our timeline of breeding and fecal sampling in figure B were the respective colonic microbiota were assessed over 3 generations from parental to F1 to F2, and then over a span of 5 months. And the boxes just illustrates the timeline when fecal matter from the different mice were used for 16S RNA gene sequencing. And then in the next two slides, I'll share with you some data that we collected. So firstly, we assessed the colonization efficiency by calculating in each generation at selected age. See it in figure A on the leftmost column that tells you of either P, F1, F2, and then different ages. And then within each different time points from each different either parental F1 or F2 generation, we identified the number of taxa on the genus level in those recipient animals. We determined the fraction compared to the taxa of the inoculum, meaning from the black C57 black 6 NTEC mice. So for example, in row 1, the colonization efficiency of parental mice, P mice, determined 7 days after inoculation was 48% in the isolator and 51% in the IVCs respectively. And then in the last two rows, for example, the colonization efficiency of F1 and F2 mice at different ages, at either 18 weeks of age or 11 weeks of age, it was definitely higher than the parental mice. 7 days also after inoculation in the isolator, so 68% in the isolator. It's a little bit higher than that in the IVCs, 65% approximately. However, when we did the statistical comparison, there was no significant difference found in the colonization efficiency between the two different housing systems at any time point, right? So meaning when we compare isolator versus IVC at each horizontal row, there was no significant difference. Then in B, we looked at the number of overlapping taxa compared to the inoculum of all samples, and we assessed whether more or less taxa we detected in the less protected IVC housing system compared to the isolator. So a total of 53 genus level taxa were identified in the inoculum, and of these, 47 were recovered from the mice in the isolator, circled in the red, compared to 48 in the IVC, circled in blue. And there was 45 common between all three. So, and then in the bottom figure C and D, to further address the hypothesis that IVC housing may or may not significantly affect the number of taxa, we looked at alpha diversity, which is just a measure of the microbiome diversity applicable to a single sample. And in this case, we use two different indices. The richness index in C quantifies how many different types the data set of interest contains, and the Shannon index is commonly used to characterize the predictive uncertainty of the data set. But overall, either from a forward-looking perspective or reverse-looking perspective, there was no statistical significant differences using either the richness index or the Shannon index in the alpha diversity metrics between the two different housing systems at any age, at any time point. And then in the next slide, we then, after assessing alpha diversity, we proceeded to assess the beta diversity of the gut microbiota. And beta diversity is a measure of the similarity or dissimilarity between the two microbiota communities. And in this case, we firstly wanted to investigate between the generations of the mice at various ages from each housing system, and using unweighted unifract principal component analysis plots. This is a visual appreciation. So in figure A, whether we definitely do not visualize any distinct clustering between the different housing systems from between the different generations, not visible. And then in B and C, we now have a deeper dive into the beta diversity of the gut microbiota of isolated and IVC housed mice over 3 generations, where in B, they are fecal samples from F1 mice aged 4 weeks only, and in C was fecal samples from F1 mice aged at 11 weeks. And we find there's a low, there's statistically significant separation, but low in figure B between IVC versus isolators at 4 weeks of age. But when they grow up older to 11 weeks of age, there's no significant separation then. So there are some additional interesting supplementary information in this this publication. But from these figures that I shared, the general conclusions are that the IVC system definitely can provide a level of protected housing for neurobiologic mice, because the donor bacterial community was equally represented in both IVC and isolated housing systems. And we do detect, for example, in this slide, that there's a time-dependent clustering between generations. It can be observed, but the temporal composition, the dynamics of temporal dynamics of bacterial composition was definitely stronger in the IVC housing system. And we understand that complex microbiotas can be protected in an IV system, but challenges still remain, such as I mentioned at the time, the temporal dynamics, which should be addressed, I think, moving forward as a future project, because they seem to be a bit more pronounced in IVC house systems, and we should better understand in the discussion also later on. We can further discuss how we can proceed forward based on the detailed methods and methodology that we described in this publication. So I pass the next slide to Sylvie.
Yes, thanks, Ivan. So now I will present you some case studies related to a pathological situation and in which different FMT applications are described. And we will start with the avatar model. That may be the best approach to transpose the clinical situation into a preclinical study. So indeed, the avatar model can recapitulate the patient phenotype in 2 recipient mice upon fecal microbiota transplantation. In other words, it means that in case of a non-responder cancer patient, the resistance to PD-1 blockade is transferred into recipient mice. And in fact, this approach allows them to evaluate the capability of a product to rescue the anti-tumoral efficacy of the immune checkpoint inhibitor and to identify biomarkers of efficacy in such context. So upon environment request, we have put in place such approach to meet their needs, using here a mouse fibrosarcoma model engrafted on SPF mice, pre-treated with a cocktail of broad-spectrum antibiotics for FMT purpose. And as shown in this example, the transfer of a stool from a non-responder cancer patient tended to reduce the efficacy of the anti-PD-1 antibody as compared to abiotic mice, in which an efficacy of the anti-PD-1 was reported. But interestingly, the oral supplementation with bacteria X but not Y in such context was able to rescue the efficacy of the anti-PD-1 and at a level that was comparable to what is observed in abiotic mice. So DFMT can be used also as a treatment by itself and in pathologies other than cancer, such as inflammatory disease, and here colitis. And in this study case, the daily transplantation of fecal material coming from an healthy rodent into mice with colitis induced by DSS led to a faster recovery when compared to untreated animals, as reported by the disease activity index. And in fact, this scoring is taking into account body weight loss, stool consistency, and blood in the feces. So the FMT was able also to reduce the colon inflammation as revealed by following parameters: the weight length ratio of the colon, the epithelial thickness of the colonic mucosa, and also the cytokine expression in colon tissue and here in distal part. So in fact, together, these results demonstrated the capability of FMT to therapeutically control colitis and pose also FMT as a valuable therapeutic option in immune-related pathologies. So in conclusion, different strategies can be conceived to get efficient engraftment in order to demonstrate the benefit of FMT on health, but also to recapitulate properly the phenotype of a donor. However, the researcher need to carefully consider the goals of their studies to determine the most suitable approach. And as you know, up to now, no standard protocols for FMT procedures exist. And this should be for sure a priority for having reproducible data. For sure, this need of standardization is also true for other microbiome-derived therapeutics. So today, we focus only on a single microbiome-derived approach, that is a fecal microbiota transplantation. For sure, there are so much to say on this topic and other approaches that may be of interest to you as well. So unfortunately, due to lack of time, it was not possible to present all these points today. But don't hesitate to contact Ivan and me for any question and request you may have. So we thank you for your attention. And Sonia, I think that this is time for a new poll session.
Yes, it is, Sylvie. First, I'd like to say thank you very much to Sylvie and Ivan for that very insightful presentation. I hope everyone enjoyed it. We're going to run 2 quick poll questions before we get into the Q&A portion of the webinar. So audience members, just like before, the screen will take over, and there you go. There's poll question number 2. So please go ahead and cast your vote on this poll question here. I'll give you 40 seconds to complete this poll. Then we'll go into poll number 2. So, 3, I should say. So poll number 2, we have you already performed FMT experiments? And if so, what was the application? So you have no, yes for evaluating FMT as a therapy, yes for recapitulating a donor phenotype, or yes for both. So please go ahead and cast your votes. I am truly appreciative of your participation in these poll questions. Oh, perfect. That looks like we have the majority of our audience members have cast their votes. Just remember, just click on any of the below items that suits you. And then I will close the polls and share the results. Okay, thank you everyone for participating in poll number 2. Let's take a look. Okay, so here are the results for poll number 2. We have 94% of our audience members saying no. Yes, or we're looking at 0%. Then we have 6% saying yes for recapitulating a donor phenotype, and then 0% for yes for both. So thank you everyone for participating in poll question number 2. Now let's go right into poll question number 3, our final poll. And there you go. And the question we have for you is, which type of rodent model would be of primary interest to you for an FMT experiment? So you have 1, 2, 3, 4, 5 options below to choose from. Germ-free animals, antibiotic-treated animals, laxative-treated animals, or in-house animal model, but the final option there is other. So please go ahead and just select on one of the below, and then I will close the polls in a few seconds once we have the majority of our audience participating in this poll question, and then I will share the results, and then we'll get right into the Q&A. Okay, that looks like it's the majority of everyone. Thank you so much. Let's take a look at the final results of our final poll. Okay, so here are the results. We have 50% of our audience are saying germ-free animals, then 25% are saying antibiotic-treated animals, 0% are saying laxative-treated animals. We have then 19% in-house animal model, and then just 6% of our audience members are saying other. So thank you everyone for participating in poll number 2 and 3. And now we're going to begin the Q&A portion of the webinar. So I'll invite Sylvie and Ivan to join us here. And remember, audience members, to send in your questions by using that questions window that is located on the right-hand side of your screen. And we'll try to attend to your questions during the time that we have together. So Sylvie and Ivan, are you ready for our first question?
Yes.
Okay, all right, let's take a look here. Okay, I have here this audience member asking, based on your experience, what is the most critical step for having successful engraftment? Who would like to answer that one? Sylvie or Ivan, who would like to answer that question?
Would you like to start?
Yes, I can start. In fact,
Okay.
I would say that, in fact, all steps during FMT are critical. So from the qualification of the donor samples to the analysis of the data. I think that Ivan has well described all key steps and all critical steps. So for sure, it's important to track all details of the protocol at time you do an FMT experiment to understand what happened during your experiment, and also to be able to come back to these details in case you will not get, I would say, the expected results or regarding your FMT experiment, and especially a successful engraftment. So I don't know, Ivan, if you want to add some information.
Yes, I totally agree that everything everything with the key characteristics and considerations highlighted in some of the case studies that we shared today. They would all affect the successes of your FMT experiment. And what Sylvie and I mentioned multiple times during this presentation is your hypotheses driven by the goal of your project is key. This is something that with some researchers, they clearly know what they would like to achieve. On top of that, they know after their hypothesis 1, they know what the hypotheses 2 and 3. So it's not just planning for one project or one experiment, but understanding your overall, in this case, for example, the pipeline or discovery program. This really helps, as what Sylvie mentioned, designing something that would be replicable and reproducible subsequently. Yeah.
Okay, thank you very much for answering that. Let's go on to question number 2. This audience member is asking, what are some non-invasive and invasive procedures for collection of fecal matter, and how could a researcher decide between these? We'd like to start with that one.
Ivan?
I could, yes. Yeah, definitely. So as what Sylvie and I, our focus is really the clinical aspects and the reiterative preclinical design. So I start off with what we know, the non-invasive and invasive procedures, starting with in clinical human patients, right? Adults, healthy adults, or clinical patients. It's feces is most commonly used. Is the most non-invasive procedure. But in humans, we also would perform endoscopic sampling. It occurs sometimes, and in endoscopic, it typically is more proximal to the rectum, so more colonic, more rectal types of sampling. And then, as what Sylvie has mentioned, those kinds of samples have been successfully used in either clinical management strategies or clinical studies, and or also reverting to a preclinical experiment and project. So sometimes when we want to model the clinical condition, yes, we also like to see, okay, should we just collect fecal matter from the bedding, and or we do a rodent endoscopic procedure. So that is non-invasive. But understandably, if it's endoscopic, you need trained surgeons and animal care technicians, importantly also we require the appropriate animal ethics and protocols. And in this case, this is a non-invasive and or invasive procedure that is not terminal. Means it can be longed. Animals can be longed. We can do longitudinal sampling. However, in FMT experiments, there may be arguments for an invasive procedure that may be terminal, because in humans, yes, we can do endoscopy either from an oral gastric procedure or like an OGT setting, and we can even sample up to the proximal small intestines. It's possible. But in a rodent, it'd be very challenging, and successes aren't high. So if you want to sample things from the rodents, like the jejunum and ileum, researchers will perform terminal experiments where they have to euthanize and just collect it from the gut itself. Yeah. So these are some commonly used either invasive or non-invasive procedures for the gut.
Okay, thank you very much, Ivan. I'm going to squeeze in one more question for this Q&A before we close out. This audience member is asking, how should stool samples be stored to minimize the impact on the microbial survival? Who would like to take that one? Sylvie or Ivan? Did you hear that one?
No, I'm sorry. I think there's some static.
Oh, no problem, no problem. I'll go right back to that question again. The question I have here is, how should stool samples be stored to minimize the impact on the microbial survival?
Oh, right, the storage of the, okay. That's a pretty important one. Mm-hmm. Yeah, as I said, the choice of, it really is driven by your aim and the goal. And very importantly, also, what is your source? What is the donor? Do you have the ability to return to your donor, be they from humans or from rodents? And that would influence whether you would want to think about either doing it fresh transplant, or if you have limited access to the donor material, you might have to consider having to freeze them or preserve them in some way. And that is one of the most important consideration when it comes to storage of the transplant or processing. Others would be cryoprotectant. I know that there have been papers showing that cryoprotectant, the addition of cryoprotectant may affect the microbiota recovery. So that is one key consideration that you have to consider. I mean, in before doing any FMT, just to do the process of recovery, freezing, and then comparing if there is any change in the microbiome status. In terms of dilution media, currently in a clinical setting, whether the commonly used dilution media, if it affects the success of the FMT treatment, there doesn't seem to be a significant difference. So it's still, as what Sylvie has highlighted, the methodology and the publication and the description, a lot of groups still would utilize many different processes and protocols.
Okay.
Sylvie, would you like to add anything?
Before we shut down here, before we close
yes, the only thing that I would like to add is, anyway, if you plan to work with frozen sample, you have to be sure that they work, that you have a similar result when you use this sample as fresh, in fact. And this is one of the example I presented today, in fact, in case of colitis treatment with FMT. And from our side, we have checked that the condition in which we store the stool and that we freeze the stool, at the end, when we use them for the treatment, we obtain a result that was similar to the one obtained with the fresh sample. So this is important that to double check at this point as well.
Okay, well, thank you very much, Sylvie and Ivan, for answering all those questions. We have come to the end of the Q&A portion of the webinar. If we couldn't attend to your questions, the team at Oncodesign may follow up with you after this presentation. If you have any further questions, please direct them to the email addresses that are on your screen, and there's also phone numbers there. And then you see there's also their, where their headquarters are located. So please go ahead and reach out to them. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now in a few seconds, I'm going to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues once they register for the recording as well. So I encourage you to do that. Now please join us in thanking our speakers, Sylvie and Ivan, for that very insightful presentation. So thank you again, Sylvie and Ivan.
You're welcome.
Pleasure.
Thank you.
Thank you. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at Xtalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye everyone. Thank you.
Bye.
Thank you. Bye.
Take care.